Hello students, welcome to EPG Pathshala. I am Dr. Nidhi Garg from Deshbandhu College, University of Delhi. Today we are going to talk about large-scale analysis of genome, functional genomics from the paper Molecular Genetics. After studying this module, you will be able to annotate genes in newly sequenced genome. You will be able to ascertain the function of unknown genes for which we don't know the phenotype yet. You will be able to find evolutionary relationship of genes with other genes, and you will be able to identify the functional role played by an organism in the community, followed by the brief summary of this module. The entire genetic content of an organism is known as its genome. Genome annotation or functional genomics is a branch of science that deals with the identification and characterization of the genetic content of an organism or organisms to assess the function played by that organism within the community. This includes organisms from bacteria to humans. Now, the organization of genome is quite complex and it consists of both protein coding as well as non-coding sequences. The protein coding sequences are popularly known as genes, which constitutes information for particular traits, say my height, skin color, eye color, hair texture, type of blood group, etc. Whereas the non-coding sequences do not code for these proteins, but they're equally important because they regulate the development, albeit in different ways. The nuclear genome is divided into 24 linear chromosomes. Now, these 24 chromosomes comprise of the 22 autosomes and 2 sex chromosomes, that is the X and the Y chromosome. It is made up of 3 into 10 to the power 9 nucleotides. The human genome encodes about 20 to 22,000 genes. Now, the mitochondrial genome is present as a circular form within the mitochondria. It is made up of 16,569 nucleotides and it has very few genes present in multiple copies. The process of decoding DNA into useful information comprises of two processes. First, transcription, then translation. Transcription is the process wherein information in DNA is copied into messenger RNA. During expression, the mRNA moves to the ribosomes and acts as an instructor to determine the order of the amino acids within the protein, the phenomena known as translation. The unidirectional flow of information from DNA to RNA and from RNA to protein is known as the central dogma, a term that was coined by Francis Crick way back in 1956. Another level of complex regulation is that expression of all the genes is not uniform, and not all genes are expressed in each cell at all times. There is differential expression of genes at the particular time in a particular cell. Therefore, gene expression is a tightly regulated process. It is different and unique gene expression which differentiates a cell, say a cell that is a muscle cell or a skin cell or a neuronal cell. Because of recent advances in sequencing technology, it is now possible to sequence the complete genome. In fact, the genome of many model organisms such as yeast, Drosophila, mouse, etc., and interestingly our own genome has been sequenced. The new branch of science named Genomics has emerged, which majorly deals with, first, the mapping and sequencing of genomes of different organisms, locating genes within the genome, and predicting areas of non-genic regions within the genome. The study of genomics is very informative and has several applications. One of the major challenging tasks of genomics is functional genomics, which majorly deals with identification and annotation of the genes in an organism to assess its role in the community. This includes annotation and assigning function to genes, to analyze which genes are expressed in a particular cell at the particular time and their level of expression, to identify whether all expressed genes translate into final products, that is protein or RNA, to detect different protein profiles in different cells and in same cell at different conditions. Based on how the above questions are answered by functional genomics, the study has been divided into three parts. First, annotate and assign function to a gene. Second, analyze the mRNA transcript, or we call it as transcriptomics. Third, analyze and identify the protein profile, also known as the proteomics. In this module, we will be majorly focusing on the role of gene annotation and assignment of function to a gene. The concept of transcriptomics and proteomics will be dealt in detail in subsequent modules. Methodologies for gene annotation. At the beginning of genomic era, gene annotation was restricted to a single gene or a small plasmid or a kb region of DNA. Initially, the researchers used to copy and paste individual gene or DNA sequences for annotation in the blast of NCBI. This approach is feasible if there are a few gene sequences to be annotated. But if one has to annotate the complete genome of an organism, this approach is impractical. With the advancement of functional genomics, large number of softwares are available that can annotate the total genes present in a genome or number of genomes and even metagenome. The annotation softwares or the tools are either available online or can be downloaded, which can be run on the Linux or Windows or Mac operating systems. The following is the list of genome annotation softwares. Most importantly are the NCBI Prokaryotic Genomes Automatic Annotation Pipeline, known as the NCBI PGAP. Second is the Rapid Annotation Using Subsystem Technology, or the RAST. Third is the Institute for Genome Sciences, IGS. A few others are as follows: Bacterial Annotation System, BISYS, Maker Web Annotation Service, MVAS, Genome Sequence Annotation Server, GenSAS, ORI Finder, followed by Metagenome Rapid Annotation Using Subsystem Technology, known as MGRAST. We will be talking one after the other about the different annotation softwares. First of which is the NCBI PGAP. With genome submission at NCBI database, they provide the GenBank file of your bacterial genome, which is considered as the most authentic sequence annotation. It provides sequence information in the form of both nucleic acid and protein sequences, their coding direction, their location, presence of tRNA, rRNA, pseudogenes, and the total coding sequences. It is available at the following link. Rapid Annotation Using Subsystem Technology, RAST. It is an automated annotation system that provides a user-friendly interface. Users can just upload their data in the form of contigs or scaffolds or replicons, and it can provide the annotation report along with the complete sequence of CDS and other general genomic characteristic features. It can also build the metabolic model of the organism to identify the complete functional role. In addition, Seed Viewer provides the enhanced view of the subsystems and distribution of genes. Users can also compare their data with the other available or the uploaded genomes. The data information remains stored for future reference of the user. The link is available at the following link. Institute of Genomic Sciences, IGS, the Prokaryotic Annotation Pipeline. It is an automated pipeline for annotation of both bacterial and archaeal genomes. The protein prediction methods mainly include the HMM search, pairwise alignments, and the multiple motif prediction. Along with protein coding information, non-coding RNAs can also be predicted by the pipeline. Results can be viewed using the Manatee Annotation Visualization tool and can be obtained in the form of different multiple file formats. To upload or annotate the data, the user has to contact them via the email. It is available online at the following link. The Bacterial Annotation System. It is a web server for annotation of bacterial genome sequences. It uses more than 30 programs to identify nearly 60 subsystems and designate each gene according to their respective subfields. It also predicts the possible orthologs and paralog sequences. Users can also generate the circular view of their query chromosomal and plasmid sequences with the gene annotation. These annotations can be downloaded and viewed by the respective user. It is available online at the following link. Marker Web Annotation Service. It is a web-accessible genome annotation pipeline. It allows the annotation of small or intermediate genomes of prokaryotic and eukaryotic organisms. Users can analyze their data and produce output to be loaded on genome databases. It is available at the following link. Genome Sequence Annotation Server. It provides a graphical interface to obtain multiple structural and functional annotations. Users can upload sequences and can map ESTs, identify repeat regions along with the gene functions. This also provides different file formats to the users which can be utilized for further analysis at different tools. It is available at the following link. ORI Finder. In order to identify the ORI-C region in bacterial genomes from where the replication starts and considered as the first nucleotide sequence, users can use this online available tool. It identifies base composition asymmetry and distribution of DNA A boxes and predicts the ORI region. It is available at the following link. Metagenome Rapid Annotation using Subsystem Technology. It is an automated server for annotating data directly from environmental samples. It can also assist in phylogenetic classification and metabolic model construction. It is available at the following link. Example of genome annotation and comparative pathway mapping. In a study, comparative functional profiling was performed on bacteria belonging to genus Fingobium. This group of organisms have been predominantly known for their ability to degrade the xenobiotic contaminant hexachlorocyclohexane or HCH. The annotation obtained from RAS server was used for further building the comparative metabolic profile among the 9 Fingobium strains. This profile is further visualized in the form of a dual dendrogram, and the strains were clustered on the basis of their top 50 metabolic families. The following conclusions were drawn from the analysis. First, the phylogenetic relationship among the strains on the basis of their metabolic dominance was identified, such as strains which, however, shares the close nucleotide identity were clustered separately in their metabolic profile. Secondly, using color gradients of the heat map, the abundance of each pathway with respect to each strain was determined. For example, 1,4-dichlorobenzene degradation is predominated in strains LL02, P25, HDIPO4, UT26 and B90A, while glycosamine glycan degradation is predominantly marked in strain RL3. Methodologies for understanding gene function. As more and more genome sequencing projects are completing up, through gene annotation, new genes have been identified at a faster rate. For example, when gene annotation study was performed on complete genome sequence of E. coli in yeast, more than 50% of the genes were identified and characterized which were not previously known by classical gene analysis methods. There are two major approaches to find out the function of an unknown gene. First is the computer-based method and second is the experimental approach. Computer-based method, homology search. Homology searching is based on the assumption that the related genes have similar sequence. This method is also known as similar sequence research. For example, human hemoglobin and mouse hemoglobin are evolutionarily related with similar sequence. Now the next question arises, how to perform homology search? Here, the sequence of the unknown gene or the query gene can be compared or aligned to sequence of genes in the biological database. Computer-based method, homology search. Now, if the query sequence and the subject sequence are similar, then we can predict that both the genes are evolutionarily related or homologous, and find the function of the unknown gene which might be the same or at least similar to the subject gene. Homology searching can be done with a DNA sequence or even a tentative amino acid sequence. The amino acid sequence is then compared to the amino acid sequence of different gene in the biological database. But one thing needs to be noted that comparing an amino acid sequence is considered better than DNA sequence because there are 20 different types of amino acids in a protein as compared to just 4 nucleotides in a DNA sequence. So there is comparatively less chance of random alignment as compared to a DNA sequence. Now, when the query sequence is aligned with the subject sequence, a score is calculated, either based on the number of identical residues, which gives the degree of identity, or based on the number of similar or closely related residues. When the residues are chemically related, say leucine and isoleucine, which gives the degree of similarity. Then, based on similarity score, the homology between the two sequences can be inferred. Sometimes during alignment, gaps may be included to have best overall score. Gaps in turn simply imply the process of, say, the insertion or deletion of nucleotides from a gene during the course of evolution. The homology search can provide information on the function of the whole gene or maybe a part of the gene. Several computer software programs exist for sequence alignment. The most popular are BLAST, which is the basic local alignment search tool. It is available at the National Center for Biotechnology Information at the NCBI website. The BLAST accepts a query sequence and searches the best probable match in the whole database for either a whole sequence or part of a sequence of query sequence. Maximum softwares used for gene prediction look for particular DNA sequences for identifying a probable gene in the DNA sequence or genome. These sequences should be encoding a protein and are known as the open reading frames. An ORF generally starts with the AUG codon followed by other codons that specify the protein's amino acids and ends with a stop codon, which may be UAA, UAG, or UGA. The ORF region in the DNA sequence is identified by softwares by using overlapping reading frames of the 3 nucleotides each. Let us take, for example, the sequence ABCDEFGHIJK, which can be read as 3-letter words in the following ways: ABCDEFGHI, or BCDEFG, or HIJ, or CDEFGHIJK. These softwares quickly scans the DNA sequence or the genome using the overlapping reading frames on both the original strand as well as the complementary strand, generating 6 different reading frames for every DNA sequence. Using these gene prediction programs, ORFs can be easily searched in the bacterial genomes as the DNA sequence matches the mRNA. It is tough to find genes in the genome of the eukaryotes because the gene contains the coding regions called exons interrupted by non-coding regions called introns. Following transcription, the mRNA undergoes post-transcriptional processing during which the introns are spliced or removed, generating a functional mRNA that encodes the desired protein. This final spliced mRNA is smaller in size than the original RNA transcript that matches the DNA sequence in the genome. In addition to this, there exists the phenomenon of alternative splicing, where the same mRNA undergoes splicing such that not only the introns are removed, but the exons are also removed in different permutations and combinations, generating many different mRNA transcripts. These final spliced mRNAs give rise to different proteins. This is responsible for the huge diversity of proteins in the humans, although their genome contains just about 24,000 protein coding genes. Thus, a simple ORF finding program is unsuitable for searching genes in the genomic DNA of eukaryotes, as they contain introns in their genes which do not match the mRNA. There exist several advanced sequence analysis programs which, in addition to simply identifying ORFs, use other information to identify genes in the eukaryotic genome. Limitations of homology search. It is not always true that homologous genes will have similar functions. Sometimes homologous genes have different biological functions. For example, gene for crystallins of the eye lens is similar in sequence with the gene for metabolic enzymes, but both have entirely different function. So, if the given query sequence has come similar to the sequence of the crystalline gene, the function might not be same as crystallins of the lens. It might be a metabolic enzyme and vice versa. There are cases when genes are not similar in sequence but still have the same biological function. It might be possible due to sharing a part of gene which might be encoding for a protein domain which is responsible for same function between unrelated genes. For example, a gene segment encoding the Tudor domain is present in evolutionary unrelated proteins. It is responsible for the shared function, that is, interaction between protein and RNA. Sometimes the function of a gene or protein in the database is not known. In that case, even if homology is found, but still the function of query gene is not found. Those query genes are known as function unknown genes or FUN, and such genes and their homologues come under orphan families. Sometimes there is no homologue of a query gene in the database. Such genes are known as single orphans. Although homology search methodology is most commonly used to find out the function of unknown gene and is a fast method, but there is a need to confirm the exact function of the gene, so experimental analysis is used. Experiment-based approach. Experiment-based approaches are used to complement and extend the results of homology-based research analysis to ascertain the function of a new gene. It is the most challenging job as the experimental-based method follows reverse route of conventional genetic analysis methods. Here, the starting point is gene, and the objective is to identify its function or its effect on the phenotype. This involves mutating the gene and follows its consequence in phenotype to find its function. All the strategies used in this analysis are not equally applicable to all unknown genes. Thus, for assigning function to a gene, the experiments can be performed using DNA, RNA, or protein. We will be first talking about experimental approaches using DNA. One of the easiest approaches is the gene mutation, where a target gene can be altered by either loss of function, that is gene inactivation, or by gain of function, that is gene overexpression. Gene inactivation. The gene whose function is to be determined is inactivated and its function is ascertained. This can be done using two techniques, that is, by either using homologous recombination or by transposon tagging. Homologous recombination. In the technique of homologous recombination, the sequences bordering the target gene is incorporated in the vector DNA along with the non-specific gene. The vector is mixed with the DNA containing the targeted gene. There will be a homologous recombination between the vector DNA and the target gene due to the process of homologous sequence in the vector DNA. Thus, the target gene is replaced by a DNA segment which disrupts its function. If one has to find out the consequence of target gene inactivation, the knockout mice are produced, where the phenotype of the mice clearly shows the function of the targeted gene. It is the most common strategy used for determining the function of a gene in eukaryotes, particularly genes related to disease conditions such as cancer. In mice, the embryonic stem cells are used and engineered via homologous recombination method. Embryonic stem cells are taken from the inner cell mass of blastocysts. They are pluripotent cells that can produce all cell types and can also be grown in culture. In these stem cells, vector DNA is introduced either by electroporation or microinjection, where homologous recombination takes place. The DNA fragment inserted in place of the targeted gene contains an antibiotic-resistant gene, through which transformed embryonic stem cells are selected in culture. These cells are injected into the inner cell mass of blastocysts, which are transferred to newly mated pseudo-pregnant females. The mouse offsprings are tested for the presence of gene by removing a piece of tissue from the tail by PCR. About 10 to 20% of mouse offsprings will have the gene and will be designated as chimeric mouse, as they contain a mixture of non-altered cells and altered cells. The chimeric mouse is allowed to mate with another chimeric mouse so that 1/4 of the offsprings will be homozygous for the inactivated gene. The particular mice will be termed as the gene knockout mice. The phenotype of such mice will provide desired information about the function of a target gene. Limitations of homologous recombination. Now, this technique somehow works best for many genes, but the major limitation is if the gene is important for the organism's survival, especially during development, then it cannot be studied in homozygous knockout mouse, because in this case, both the copies of the gene has been knocked out. Secondly, the knockout mouse are sometimes unaffected by the inactivation of certain genes. Maybe certain genes are not so vital for their existence. Thirdly, we may say that the mouse genome is sufficiently redundant to compensate for the gene loss. Fourthly, maximum genes are pleiotropic. Therefore, they are expressed in different tissues differentially and during different stages of development. Transposon tagging. The transposable element known as the jumping genes can be used to know the function of a gene, as it has a tendency to insert into any gene, making them inactive. Now, the major limitation of transposon tagging is that the transposition event is random. It is actually quite difficult to track where exactly the transpon will insert after it has jumped. Therefore, this method is more suitable for global analysis of genome, where genes are randomly inactivated, followed by examining the progeny for phenotype changes. Gene overexpression. Not only loss of functions, but sometimes gain of function or gene overexpression will also help us to find out the function of a gene. To overexpress a gene, a cloning expression vector is required, which has tendency to maintain itself in high copy number. The target gene is cloned along with a strong active promoter in cloning vector and expressed in high copy number. The engineered cloning vector is introduced into the host, and the host with large copies of target gene will be transgenic for the particular target gene. The engineered cloning vector is introduced into the host, and the host with large copies of the target gene will be transgenic for that particular target gene. Using this method, the researcher has to be very cautious whether the change in phenotype is due to the specific function of the target gene or it is a consequence of abnormality due to overexpression of target gene. Phenotypic effect of mutation of gene. After gene inactivation or gene overexpression, its consequences are ascertained by identifying a change in the phenotype. This step is very difficult and complex, as there are immense range of phenotypes to be examined. For example, in yeast, typical phenotypes to be assessed are DNA synthesis and the cell cycle, RNA synthesis and processing, protein synthesis, stress response, lipid metabolism, DNA repair and recombination, protein trafficking, development, and meiosis. In multicellular organisms, it becomes even more complicated. Experimental approaches using RNA. Microarrays. Microarrays are used for identifying the patterns of gene expression under a particular set of conditions or in a process. For example, one can differentiate between the pattern of gene expression in a normal cell and a cancerous cell. There will be variation between the transcript levels of these two different conditions. Then, one can compare the results obtained with the expression pattern of genes with the known functions. Microarrays basically measure the quantity of total mRNA in a given sample. The total mRNA is isolated from the given cell type and reverse transcribed into cDNA and fluorescently labeled, which are then hybridized to the probe DNA. The probe DNA sequences are immobilized on a solid glass surface, which is the DNA microarray. The intensity of fluorescence of a spot on the microarray is directly proportional to the amount of mRNA in the sample and thus gives measurement of the gene expression. SAGE, which stands for Serial Analysis of Gene Expression. It is another method by which analysis of gene expression can be done. This method involves sequencing of the total RNA isolated from the cell instead of hybridization, as we have seen in the case of microarray. In SAGE, 10 to 17 base pair tags are sequenced as they are unique for each gene. These unique tags are produced from poly-A mRNA and then ligated end-to-end before sequencing. SAGE offers the advantage of giving an impartial measurement of the number of transcripts per cell because, unlike microarray, it is not dependent on former information of what transcripts to study. RNA interference. RNA interference provides the indirect approach of gene inactivation as it acts on mRNA of target gene and prevents the expression of target gene. The interfering RNA are produced in the cell by introducing into the cell short, double-stranded RNA molecules which are complementary to sequence of target mRNA. These double-stranded RNA molecules are broken down into single-stranded interfering RNA which induce degradation of target mRNA. C. elegans and Drosophila are the organisms of choice for the application of RNA interference for studying functional genomics, as RNA interference is most effective in these organisms. C. elegans is principally used for RNA interference research due to two reasons. First, the effect of gene silencing are usually heritable. Secondly, it is very simple to deliver the double-stranded RNA to this organism. Bacteria like E. coli containing the target double-stranded RNA are fed to C. elegans, which through a lesser known mechanism transfer the double-stranded RNA to the intestinal tract of the worm. This is known as delivery by feeding, is equally effective in inducing gene silencing in comparison to expensive and time-consuming delivery methods like the soaking of C. elegans in solution of double-stranded RNA or injecting of double-stranded RNA into the gonads. In comparison to C. elegans, the delivery of double-stranded RNA is more difficult in most of the model organisms, but the researchers are making effort to carry out large-scale genomic screening in cell culture with mammalian cells. In case of plants, RNA interference is a principal technique used for both genomic mapping and annotation. Reason being that most plants are polyploid. Therefore, the traditional genetic engineering techniques are insufficient for functional genomic studies. For example, in bread wheat, Arabidopsis and maize, the use of RNA interference has been successful. Experimental approaches using proteins. Yeast 2 hybrid system. This system is used to test the probable interaction of a protein called as the bait against several proteins known as the prey. The main aim is to study physical protein-protein interactions. It is possible to test a library of bait proteins against a library of prey proteins for identifying all the likely interactions within the cell. This screening system was devised on the basis of a transcription factor called GAL4. The GAL4 has two distinct domains. Firstly, it has a DNA binding domain. Secondly, it has a transcription activation domain. Both the domains are required for GAL4 to initiate the transcription of a reporter gene. In the yeast 2 hybrid strain, the bait protein whose function is to be determined is fused to the binding domain of GAL4. A library of possible prey, that is interacting proteins, is recombinantly expressed in a vector fused with the activation domain. Now, in vivo interaction of bait and prey proteins within the yeast cell will bring the activation and binding domains of GAL4 close enough, leading to the expression of a reporter gene. Thus, the interacting bait and prey proteins can be isolated and characterized. Affinity purification and mass spectrometry (APMS). This technique is used for identifying proteins that interact with one another in complexes. A bait protein is used and a complex of proteins is allowed to form around it. The identification of the bait protein is done by using either an antibody against it or through a recombinant tag, as it allows extraction of the bait along with the proteins in the complex. The isolated proteins undergo digestion, yielding short peptide fragments. The proteins are identified on the basis of their mass to charge ratio by mass spectrometry. Limitations of experiment-based methods. These strategies are not equally applicable to genes of different organisms. For example, gene inactivation by homologous recombination works best for yeast, but its efficiency is poor in case of mice. Screening for change in phenotype is very difficult and lengthy process. Sometimes after mutation of a particular gene, there is no change in the phenotype. Thus, finding the function of the gene becomes even more tough. Sometimes strategies used for gene mutation leads to change in phenotype, which is not due to abrupt or excessive function of the gene, but which is due to changes in situation. It will indicate non-specific function of target gene. Case study: assigning the function of genes to yeast genome. Yeast, or the Saccharomyces cerevisiae, is a simple eukaryotic model system. Its sequencing was completed in 1996. Its genome organization revealed high gene density with less intergenic DNA and very few introns. The total number of ORFs are 6,274. After gene annotation, when the ORFs were analyzed, only 33% ORFs of genes were actually identified which were previously characterized. While the function of those genes was very, very clear by homology research tools, but then 67% of the ORFs were completely new. So, what to do? To know importance of those genes, there was a need to know their function. Now, the function of these new genes was assigned by experimental-based methods. The genes were mutated and the change in their phenotype was observed. Gene disruption was performed by homologous recombination and transposon tagging, followed by screening the vast number of mutants for novel phenotype that indicate the function of an inactivated gene. It gradually increases the percentage of genes with well-characterized function. So students, let us now summarize what we've learned in this module. Functional genomics is the branch of science that deals with the annotation of sequence of a gene or genome or group of genomes belonging to the same genus. This mainly helps in determining the community role of the organism and to build up its metabolic modeling. Now, with the advent of sequencing technologies and newer tools for genome annotation, functional genomics is improving day by day. Comparative functional genomics has been widely used for depicting the relative genomic features between the group of organisms which may determine their phylogenetic relationships. There are a number of tools available online, and users can access the publicly available data or can annotate their own data. Also, function of gene of interest can be identified using either computer-based or experimental method approaches. Computer-based approaches involves homology search based on the assumption that the related genes have similar functions. This method has its own limitations, so researchers try the experimental approach. Experimental-based approach involves the use of DNA through gene mutation, RNA through microarray, SAGE and RNA interference, or proteins through yeast to hybrid system, and APMS to know the function of the genes. The experimental-based methods have their own limitations. The strategies used to know the function of genes are not universally applicable. That is, it may work with one model organism but may be totally unsuitable for another organism. The screening for change in phenotype is due to mutation, is time-consuming and a lengthy process. Thank you.